The next award to be presented is the Ruth Patrick Award of ASLO 2010, which is presented to honor an outstanding contribution by a scientist that, using basic principles of aquatic sciences, addresses, informs, and solves key environmental problems. And the recipient of the 2010 Ruth Patrick Award will be Dr. Sam Luoma, and the contribution that is recognized is his work in the viability of heavy metals in coastal ecosystems, and his contribution to inform and develop new approaches to water management and water policy in California. Dr. Luoma has published about 200 papers in peer-reviewed journals and has been honored with many awards and honors, such as being named Fellow of the American Society for the Advancement of Science, and he has also been named Meritorious Senior Government Employee by the President of the United States. He is presently research professor at the John Muir Institute of the Environment at the University of California in Davis, and is also a scientific associate to the Natural History Museum in London. Between 2000 and 2003, Dr. Luoma acted as a principal investigator and lead investigator of the Calfett Bay Delta program to redesign water management in California. And with this, I'd like to invite Dr. Luoma to join me on the stage. It's a great pleasure and an honor to present you with the Ruth Patrick Award 2010, and I will read the citation that reads, "For his passionate dedication to the solution of environmental problems in San Francisco Bay and his distinguished record of innovative research, leadership, and inspirational mentoring in trace metal ecotoxicology and water resource management." Thank you and congratulations.
Thanks very much, Carlos.
Thank you.
Well, thanks very much, Carlos. That's a very nice introduction. And I have to say that, as is typical of, I think, of any scientist's career, any accomplishments in my career are the function of the tremendous people that I've worked with through time and through the decades. Some of whom you've heard from at this meeting: Dan Kane, Michelle Hornberger, David Buckwalter. Collaborations with people like Nick Fisher and Phil Rainbow, and many more people that can be mentioned. So I'd like to say that each one of them owns a piece of this award. But it's also really inspirational to receive an award in the name of Ruth Patrick. We talk today a lot about the formidable environmental challenges that we face in the years ahead. We think less about the formidable and difficult challenges that others have faced before us and actually made progress on. And I think Ruth Patrick is an inspiration to anyone who has a passion to make a difference in a world faced with these significant problems. She was born in 1907, and her first job was in 1933 at the Philadelphia Academy of Natural Sciences was as a volunteer microscopist. She was a volunteer because in 1933 it was illegal to pay women to work as scientists. She quickly turned that into a paying job and demonstrated her skills in basic sciences in systematics of diatoms, and then went on to use that basic understanding to help us understand the implications of human disturbances in streams and the effects on biodiversity. But again, that understates what was going on in that time. In fact, the streams and rivers and lakes of the developed world at that time were a complete mess. Eutrophication was widespread in lakes. Anaerobic zones were common in rivers. Whole rivers were anaerobic. Chemical contamination was at its worst in history in the developed world. New chemicals were coming into the environment. So these challenges, and there were no rules about discharging chemicals to the environment to speak of whatsoever. These are the challenges that Ruth Patrick faced. She not only used basic science to apply, to understand these problems, showing us the simplification of biodiversity that occurs in the face of chemical contamination and sewage contamination, but she also went on to make a difference in terms of policy. And this link between basic science, applied science, and policy is one that a lot of us want to do, but something that we're still not very good at, but she was good at it. She was apparently very influential in the design of one of the key pieces of legislation that made a difference in this, and that was the Clean Water Act, which was implemented in the 1970s. So today, again, we face a new generation of environmental challenges. And I think the good news is that there's precedent for addressing these and making progress. I think we absolutely have to say that that was the case with the Clean Water Act. On the one hand, I think the bad news is that the old techniques and the old approaches that we used probably have to be improved upon and probably have to be done a little bit differently. And so I'd just like to, for a few minutes today, to address maybe some of the ways that we move forward or at least continue the progress as we're moving forward. And some of these messages are messages that you've heard, I think, throughout this meeting. First of all, I'm going to talk about what kinds of science we might think about in a very broad way, and also how we should go about doing science to address these important problems. I heard Leopold, a great hydrologist and son of the environmental pioneer Aldo Leopold, talk about 25 years ago, and he said a good hydrologist is always doing three kinds of science: always keeping track of something that's going on in the environment, observing what's going on in the environment. Sometimes we call this long-term data sets. A good hydrologist is always thinking about mechanisms and processes that cause the changes that you're observing in the environment. And then there's the science that you do for fun. And he said you shouldn't get them mixed up, but all three are key ingredients. Now, not every individual is lucky enough to be able to do all three kinds of science, but all three kinds of science are essential if we're going to move forward into the future. There are far too few long-term data sets, despite the fact that we know that the solutions to our future problems lie in flexible responses and in understanding what's working and understanding what's not. But yet it's very difficult to find support for and to find time for long-term data sets. There's also a tension in the scientific community between the people that monitor and assess, and the people that do reductionist basic science. That's a long-standing kind of tension in the scientific community that I think we have to get over, because the real power in science, some of the real power in science lies in the iteration between understanding processes and understanding mechanisms, going back to nature and observing, asking questions, and coming this kind of iteration between the two kinds of science. This is an example of a long-term data set that gets back to the Ruth Patrick example, in that it shows the effectiveness of the Clean Water Act in San Francisco Bay. Starting in 1975, we've gone out for about once a month from our labs, which are over here, to this mudflat on San Francisco Bay, picked up about 30 of these bivalves, Macoma baltica, taken them back to the lab with some sediments, and just done some chemical analyses to kind of keep track of how the estuary, the idea originally was just to keep track of how the estuarine processes might influence metal concentrations in organisms. Just so happened that we were near a sewage treatment plant, discharge down here, an urban runoff discharge down here, and as the Clean Water Act was implemented from 1975, this is near monthly data, through the 1980s and into the 1990s and on into 2010, we can see the effectiveness of that rule. When we make laws like this, and they're effective laws, they can make a difference in the environment. But on the other hand, long-term data is not enough by itself, because this is a very rapid cleanup, only about a decade or so, and as the sewage treatment plant quit discharging the metals, we found less and less contamination in the clams. But this is not characteristic of all systems. In fact, if you think about, or if you know about the hydrodynamics of San Francisco Bay, it's really linked into the rapidness of this response. The bay is a shallow system with strong winds, sediments that are exchanged at the surface, and this animal feeds from the surface of the sediments. Sediments exchange from the surface annually, and so the recovery here is very rapid. In southwest England, where we've studied the estuaries, similar problems in the estuaries of the southwest, mining was a big input for metals from 1850 to 1900. Between 1900 and now, mining has gradually died off in that system, but there has been no change in the 30 years between the samples we collected in those systems in either the bioavailability of metals or in metal concentrations themselves. Again, the hydrodynamics and the sediment processes in those systems, sediment transport processes, are very different than in San Francisco Bay. And I think this brings up the second point that I'd like to emphasize, and that is our tendency to work in silos. We've heard this over and over, our tendency to work in silos and the difficulty of crossing those silos in effective ways to address our environmental problems. In ecotoxicology, we often lament the lack of ecology. In other words, we're using techniques that supposedly apply to nature, but to bridge those techniques to explanation of ecological observations is extremely difficult. In ecological, I think Emma mentioned the fact that in the ecological societies we often don't think about contaminants at all. This has been a very difficult chasm to bridge. Even though there have been many successes, it's been a very difficult chasm to bridge, and we've got to do this very much better. Why have we not been successful with that? It's not for a lack of interest, I don't believe. I think maybe the sciences haven't been ready, and maybe we haven't thought about intentionally creating the conceptual bridges. Because there are ways to create conceptual bridges, and I'd just like to show here one example of one. This is what we call our biodynamic modeling approach to understanding metal bioavailability and metal toxicity. Traditionally, the approach to understanding metal toxicity is to, and it makes sense intuitively, is to throw an organism in an aquarium, throw in different concentrations of whatever chemical you're interested in, and see what concentration the organism dies at, and that's a threshold. But of course, there are very many simplifications in that approach, and it's very difficult to bridge that approach to ecology. Our approach, what we're suggesting in this model, is that we're ready in the science now to look at all the factors that are most important in determining exposure of an organism. And exposure is the first step toward understanding the risk in an environment from a chemical. We know that the environmental concentrations of a chemical are important, but what we have less appreciation for is the importance of the physiology of the organism that we're dealing with. And beginning in the mid-1990s, Nick Fisher and I went back to the radioecologist approach and kind of deconstructed this uptake process into its various components, uptake rates from water and food, and loss rate constants, and then reconstructed with simple experiments, and then reconstructed them for the organism into a mathematical model to test what bioaccumulation would be at different concentrations. If you accept that both water and food are important as sources of metal exposure, and let's just say that it's been very difficult to get acceptance of the dietary exposure route, but I think we've shown that that's very important. Then you have to also accept that functional ecology is a really important part of determining what organisms are vulnerable to the chemical contamination, what organisms are not. The strength of a model lies in the ability to unify functional ecology, physiology, and environment into a not necessarily purely mathematical, but a combination of mathematics and experiments that can help explain exposure of organisms. Internally, ultimately, the toxicity in the organism is determined by the inherent ability to detoxify the chemical, and ultimately, toxicity is a result of that. When we took this model, our students and our colleagues have used this approach now for about 15 years. And about 5 years ago, Phil Rainbow and I put together data from 17 different studies that had used the model to predict metal concentrations in different environments, ranging from lakes to the oceans, different species, and different metals. And compared observed concentrations to predicted concentrations, and we can see that this approach, even though it's a little more complicated, it's manageable, it can be done with a variety of species, and it does a pretty good job of predicting what we see. In other words, it's a useful model. But as we all know, models are not best known or best used in just predictions. This model also helps us understand some of the basic things about the way chemicals behave in the environment and links those to both biology and ecology. First of all, this is a really interesting little story. Perna viridis is a mussel that lives in the waters of Hong Kong that Phil has studied quite a lot, Phil Rainbow. On the surface of that mussel, there often lives a barnacle, Balanus amphitrite. These animals are living in exactly the same environments. Zinc concentrations in Balanus in unpolluted waters are about 0.3% by weight. These animals are 0.3% by weight zinc. In a polluted environment, they can be 1.4% by weight zinc. In the very same environment, the mussels only have 53 to 153 micrograms per gram zinc. Interesting biological problem. If you break it apart into the flux rates, the difference is how barnacles get rid of their wastes. Barnacles have problems excreting their wastes, therefore they capture many of them into granules. Zinc happens to be one of those granules that they capture. Zinc phosphates in their body. They have a very slow rate of loss, a rate constant of loss down here, as compared to the mussel. They accumulate zinc into their tissues to very high concentrations. It's kind of an interesting biological application, but there are also very important applied applications. For example, selenium. If you heard the mountaintop mining, any part of the mountaintop mining session the other day, you heard that selenium was an issue in that. Now, if you put selenium in water and try to measure toxicity to animals, it's very non-toxic. It's very high concentrations are necessary to make a difference. But in nature, that's not how selenium works. Dissolved selenium is concentrated by the primary producers in a system to up to 1,000 to 10,000 times higher than occur in here, and that's where selenium enters the food web. The organoselenium produced by the plants is taken up by invertebrates, retained in the food web by invertebrates, or even taken up to higher concentrations and retained in the food web by predators, perhaps sometimes even magnified a little bit. But another important thing here is that the invertebrates differ because of their physiology 30-fold in terms of the degree to which they take up selenium. That means that different food webs are of different vulnerability to selenium. Here in San Francisco Bay, where we have a clam-based food web, clams with a slow rate constant of loss accumulate high concentrations of selenium. Sturgeon, which are at the top of this food web, have much higher selenium concentrations than the crustacean-based food web with striped bass at the same trophic level, where we measure trophic level by N15. That means that the first organisms to disappear from an ecosystem are going to be the organisms in this food web driven by the physiology of the invertebrates in that food web and the uptake of the plants by selenium. So it begins to help to explain why some animals are vulnerable to chemical contamination and why some animals are not. But you have to understand the food web. You have to draw that bridge between chemistry, biogeochemistry, and food web ecology. The same thing applies in stream. This is work from Dave Buckwalter and Dan Kane did in our laboratory, showing using the uptake rate constants that we talked about to calculate cadmium concentrations at a part per billion cadmium in the environment in a variety of insect species there on the left. Then we take the metabolic fraction that is detoxified in those, multiply it by the total concentration, and get the cadmium concentration in the bioactive fractions within these organisms at 1 part per billion cadmium. You can see that the difference among the invertebrate species ranges from a concentration of 30 micrograms per gram bioactive to 5,000 micrograms per gram, an amazing range amongst the insects. And what do we see in streams? And we've known this for a long time, and Will Clements has shown that in early toxicity studies that this is the case, that certain species of mayflies are the first to disappear. And this is one of the explanations of why. Not only what they eat, but their physiology is closely linked into determining the vulnerability. Species sensitivity tests that you see in the traditional toxicity testing approaches simply don't explain who does this, who disappears, and why, just simply because they don't consider all the processes that are involved. The next step then is to tie this to ecology. In this case, we're using our long-term data set from the Clark Fork River, where we've worked for about 25 years near a mining site. We're using as a measure of bioavailable copper in the river. Copper is thought to be the major problem here. Concentrations in a widespread biomonitor, hydrocyclid caddisflies. And we're looking at what we know now to be the most sensitive measure, the animals that are most likely to disappear based upon their physiology, the heptageniid mayflies. And you can see, again, that around concentrations of 80 micrograms per gram or so, maybe a little bit lower, a little bit higher, we begin to see that the abundance of the hydrocyclid caddisflies declines. Now, we can see all kinds of situations at low copper concentrations where we see more or less hydrocyclids. This is what we expect from nature. Copper is not the only thing important here. Other things can drive down populations. But when you get up to high copper concentrations, above 100 micrograms per gram in this biomonitor, you never see high abundance of hypopeptigenaids. Again, what we have is a dose response in nature using an old tool, biomonitoring, but a new concept linking ecology and bioaccumulation. An old tool linking to the ecology, the ecological structure of a benthic community. So I'd just like to say that, in conclusion, then, that it really, again, is a great honor to be given an award by this society, and especially an award in the name of Ruth Patrick. I think, again, we tend to feel a little hopeless about the formidable environmental challenges we face in the future, but I think there is precedent for addressing those. There aren't silver bullets for the solution of these, but there's a journey that we can take, and that journey will involve, again, the modifications and the improvements in the kind of science we're doing, not only within our silos, but in finding ways to better bridge those silos and think across the disciplines. Think about the discipline of the person next to you, and the more we can do that, the more likely we are to reach effective solutions. Thank you.